Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. We have a really foundational piece of longevity research to get into with you today. But before we jump into the metabolites, please take a quick second, like, subscribe, and share this deep dive with friends and family. Your support, it really sustains the Sand Science Experience and lets us keep digging into these essential breakthroughs. Okay, so today we're opening up a remarkable piece of research. It's a study that very ambitiously maps out the entire metabolic landscape of aging across the mouse body. It's called a Metabolic Atlas of Mountal Aging. And for so long, we've looked at aging through the lens of, individual diseases. We focus on one organ, maybe the liver or the brain. But the metabolic dysregulation we see with age, that shift in energy use, the breakdown of control, that's a whole body thing.
It is, and that's been the central challenge. We've been missing this comprehensive, simultaneous snapshot. We know the engine is breaking down, but we didn't really know which parts were failing first or how a failure in one part was, affecting all the others.
Right.
This study gets right at that by conducting this unprecedented profiling exercise.
And when you say unprecedented, let's really get into the scale here. They didn't just look at one or two tissues.
Oh, no, not at all. They profiled metabolic changes across 12 different organs.
12?
Yeah, we're talking the central players, heart, brain, liver, but also immune tissues like the spleen and thymus, and even specific muscles like the quadriceps and the tongue.
And across the whole lifespan, too.
The whole lifespan. They did this across 5 distinct age points, from 1 month old, which is basically an adolescent mouse, all the way up to 24 months, which is geriatric, and in both males and females.
So 5 ages, 12 organs, 2 sexes. I mean, that is just a staggering amount of biological data.
It is. And the method, liquid chromatography with tandem mass spectrometry, or LC-MSMS, let them precisely quantify 169 different polar metabolites.
And the goal here wasn't just to describe what's happening, right?
Exactly. An atlas isn't just a map of what is, it's a guide to find where the targets are. By generating all this data, the team wanted to pinpoint the pathways that actually drive aging, not just correlate with it.
So this is the foundation for finding new therapies.
It's the bedrock.
Yeah.
The absolute bedrock for identifying interventions to preserve organ function.
Okay, let's unpack this atlas. A good place to start seems to be one of the most fundamental conclusions. Aging is not uniform.
Not at all. It's a mosaic of decay, not a uniform rust. The metabolism ages differently depending on the organ and, crucially, on the sex of the mouse.
You can really see that when you look at the extremes in their data.
The contrast is dramatic. On one end, you have the thymus. That's the immune organ that makes T cells.
And it was the most volatile.
By far. A huge 44.2% of all the metabolites they detected in the thymus showed at least one significant change across the lifespan.
Which makes perfect biological sense.
It does. We know the thymus undergoes what's called thymic involution. It shrinks and just, stops working as well as we age. The metabolic chaos tracks perfectly with that functional decline.
And the complete opposite end of that spectrum.
The brain.
Right.
The brain was the metabolic bedrock. So stable. Only about 7.6% of its metabolites were significantly altered. It suggests the brain is just remarkably resilient in maintaining its metabolic balance.
Let's talk about muscle, because that's so central to aging. Sarcopenia, the loss of muscle mass, is a huge focus.
And they compared 2 very different types of muscle. The quadriceps, a big skeletal muscle for movement, was a lot like the thymus.
Very volatile.
Exactly. Over 44% of its metabolites were altered, which lines up with what we see with age-related muscle loss. But then you look at the tongue.
Which is also a muscle, a very active one.
A very active one.
Yeah.
But less than 14% of its metabolites were altered.
So the tongue muscle seems to be metabolically protected somehow.
It does. And if we can figure out why the tongue is so resistant, we might find new ways to help our other skeletal muscles.
And this all gets even more complex when you add sex into the equation.
Right. They use this visualization technique, a UMAT cluster, which groups samples by their metabolic fingerprint. And it helps you see what's the bigger driver for that organ's pattern. Is it age or is it sex?
And what did it show?
In some organs, like the quadriceps and the thymus, the samples cluster very clearly by age. The changes over time were just so powerful, they sort of washed out the differences between males and females.
But not everywhere.
Not everywhere. In the brain, the heart, and the kidney, the clustering was driven much more by sex.
So in those really vital organs, the metabolic differences between male and female are a fundamental feature all the way through life, almost independent of aging itself.
Yes, which is critical for understanding why disease risk is so different between the sexes.
Okay, that's the big picture. Let's zoom in on the specific molecules. The researchers found one metabolite that was like a universal red flag for aging,
Yes, trans-4-hydroxyproline, or HOP for short.
and it showed up everywhere.
Almost everywhere. It declined significantly in 11 out of the 12 organs they looked at.
And the exception was.
The brain, of course, reinforcing just how stable it is.
So why is this drop in free HIP so important? What does it actually do in the body?
It's all about structural integrity. HIP is a modified amino acid, and it's essential for the structure of collagen.
Collagen being the scaffolding of our tissues.
The main scaffolding, yeah. And free HIP is produced almost exclusively when old collagen gets broken down and recycled.
So if free HIP levels are dropping consistently across the body, it suggests that recycling process, it's just slowing down.
The molecular cleanup crew has gone on strike.
Right.
That's a good way to put it. This reduction in collagen degradation is a classic sign of aging, and it's what we associate with fibrosis.
The buildup of stiff, non-functional tissue.
Exactly, and they confirmed this. While free HIP was dropping, they found that older quadriceps and lung tissue actually had higher total levels of collagen. The old stuff is accumulating.
Now, you said it declined almost everywhere. There was a weird anomaly in the liver. Right.
The liver was the big outlier, and specifically in males. In female livers, HIP trended downwards like in other organs. But in male livers, it actually increased with age.
An increase that's significant.
Very. And they took it a step further. They found that if you add extra HIP to human liver cancer cells in a dish, it makes them proliferate faster.
So the age-related increase of HIP in male livers might be creating a pro-cancer environment.
It's a very compelling link, and it tracks with a higher incidence of liver cancer in men later in life.
And they confirmed this wasn't just a mouse thing.
Right. They looked at human pancreas samples from people in their 20s all the way to their 70s. And they found the same thing. The age-associated decrease in HIP was there. This is a fundamental marker of mammalian aging.
Which brings us to, for me, the most exciting part of this study. Using machine learning to find the real drivers.
The metabolic clock concept, yes.
This is so much more than just finding correlations. They built predictive models that could take a snapshot of metabolites and accurately predict an organ's biological age.
And the beauty of that approach is that the model has to identify the smallest set of metabolites that are the most predictive. If a metabolite is key to the clock, it's a huge priority for intervention.
And they found that ratios were often more important than single molecules.
Yes, the balance. Comparing metabolite A to metabolite B was often a much stronger predictor, which highlights how important metabolic balance is.
And two compounds just jumped out from these clocks. The first one is already a pretty big name in longevity circles, Alpha-ketoglutarate, AKG.
AKG is really the poster child for this right now. We already know from other work that supplementing with AKG extends health span and lifespan in different animals.
So the clock basically confirmed its importance from a totally different angle.
It did. It flagged AKG as a major driver, and then the study confirmed that, yes, plasma AKG levels are naturally lower in old mice compared to young ones.
It's a perfect convergence of evidence. The molecule that goes away with age is the same one that helps when you add it back.
But the second discovery, that was the real surprise. A total shock, actually.
Carglumic acid.
Carglumic acid. It showed up as a key driver in the clocks for the plasma, heart, spleen, lung, a whole host of organs.
And why is that so surprising?
Because of what it is. Carglumic acid is an FDA-approved drug. It's used to treat a rare condition called hyperammonemia. But until this paper, nobody knew that mammals could make it themselves. We thought it had to be an external substance.
So they were finding a drug, a synthesized drug, in healthy mice that had never been given it.
Exactly. And the levels were dropping with age. This just fundamentally changes our understanding of this molecule.
So what did they do?
They confirmed its presence in all 12 organs, and then they used some very clever tracing techniques with human cells to prove that our cells can and do make carglumic acid.
So it goes from a niche drug to a newly confirmed mammalian metabolite.
And a powerful candidate for an aging driver. It's
Yeah.
genuinely groundbreaking.
That is incredible. And the study had more of these deeper insights, too. Let's go back to the heart for a second, where we saw those big sex differences.
Yes, and those differences were really centered on redox homeostasis. So the balance between oxidative stress and antioxidants.
Okay.
The data showed female hearts maintained a much higher GSH to GSSG ratio. That's sort of the gold standard measure of a cell's antioxidant capacity. A higher ratio means better protection.
So female hearts are just better equipped to handle oxidative stress throughout life.
That's what the metabolism suggests. And it could provide a molecular reason for why women, premenopause, tend to have lower cardiovascular disease risk than men.
Fascinating. Let's pivot to another big anti-aging topic, NAD. The story is always that NAD levels just fall off a cliff as we age. What did the Atlas say?
The Atlas gave us some much-needed nuance. It really challenged that idea of a universal NAD decline.
How so?
They looked at the NAD to NADH ratio, which is a better indicator of energy status, and they found that ratio actually increased in the quadriceps and the thymus.
It went up.
It went up because the reduced form, NADH, decreased so much. The only place it showed that classic decrease was in the pancreas.
So boosting NAD might help your pancreas, but it might not be the issue at all in your muscle.
That is the exact question this data raises. It screams that interventions need to be organ-specific. What's good for one tissue might be irrelevant, or maybe even bad for another.
Finally, they even looked at differences within organs. They used a technique called MAL-D-MSI to do some spatial mapping in the kidney.
Which is an incredible tool. It lets you see where metabolites are physically located. They compared the outer layer of the kidney, the cortex, with the inner part, the medulla.
And they found localized changes.
Very localized. For instance, glucose levels went down with age, but only in the medulla, not the cortex. It points to this highly specific breakdown in how the kidney is handling glucose.
And this happened without a change in the genes being expressed.
Right. So the problem isn't that the kidney stopped making the right enzymes. The problem is happening at the level of the metabolites themselves.
Okay, so let's bring this all together. What does this massive metabolic atlas mean for you, the listener?
Well, it delivers some really fundamental new insights. I think there are three big takeaways. One, aging is tissue-specific. We need targeted therapies.
Right, no one-size-fits-all magic bullet.
Two, the decline of hydroxyproline is this incredibly robust, near-universal marker of problems with collagen. It points directly to fibrosis as a core feature of aging that we can measure.
Yeah, and three.
Three, we now have two incredibly promising therapeutic candidates that were pinpointed by these metabolic clocks, AKG, which we knew about, and this remarkable discovery of carglumic acid as a natural metabolite that declines with age.
And it's this machine learning approach, the metabolic clock, that really cut through the noise to find these causal candidates.
That's the powerful new frontier, moving from correlation to causation.
And I think the most provocative thought this study leaves all of us with is, well, it's right there with carglumic acid. Since this is already an FDA-approved drug, one we know is safe in humans for another reason, how quickly could research pivot? How fast could we start to figure out if restoring its levels can improve organ health or even extend lifespan?
The pathway to repurposing a drug is so much shorter.
So much shorter and cheaper. And that's a question for you to mull over as you think about the immediate future of anti-aging research. That's a fascinating thought to end on for this deep dive. Thank you for joining us as we mapped out the metabolic atlas of aging. And remember to like, subscribe, and share this deep dive with anyone curious about the cutting edge of science. Your support really does sustain the Sand Science experience. We'll see you next time.